Welcome to today's lecture, and today we're going to discuss how we study the human genome. One of the techniques used in studying the human genome, and actually used a lot in crime labs also. So we're going to talk about that. We're going to talk about how the DNA gets cut, how the DNA gets separated, and then how it gets read in this process for actually reading and understanding the human genome. So, the first step in understanding the human genome is to actually take your DNA, which is extremely long strands. It can be hundreds of thousands of bases long. So you're going to take each of those chromosomes and you're going to have to cut them. Okay? But we don't do it with scissors. We do it with enzymes. These enzymes are called restriction enzymes. What restriction enzymes do is they actually take and cut. They're programmed to read a certain sequence of DNA and cut it at a certain point. And each restriction enzyme has a different code that it codes for. So whenever doing this process, we use one of the restriction enzymes. It cuts the DNA at certain points, and then you can use those smaller pieces to figure out the DNA sequencing. So the code, or the enzyme EcoRe, okay, this cuts the code. It's GAATTC, and you'll notice red in the other direction also. The other strand of DNA is also GAATTC. It's going to cut between the G and the A. So we're going to make this kind of zigzag cut, and it's going to be cut into two pieces, and you're going to have two pieces that are together, base pairs are still together, and then you're going to have the four base pairs that are going to be split from each other. Okay, they're not going to be together anymore, so it's going to be one base pair together for there. The BAMI one takes and cuts. We have GGATCC, and it cuts between the two G's. So you'll notice you get the same thing here. You get those four bases that are going to be single copy or single strand. Okay. The HA3 enzyme reads for the base GGCC, and what it does is it cuts between the G's and the C's. Okay. So this one makes a straight cut, and there's no ends left over. These two, which have the ends left over, they're going to have what are called sticky ends. These four bases here are what are called sticky ends. And what that is because now we have two separate pieces, and they could fit back together, okay, but they're not going to fit back together. What they might do is actually sometimes scientists will mix two sets of DNA, and this is how they're able to do some things in the lab that aren't necessarily natural. For instance, mixing pig DNA with jellyfish DNA, and they're able to get pigs that glow in the dark with their noses and their hooves. Check it out online. They do that because they can cut the jellyfish DNA and the pig DNA with the exact same restriction enzyme. Then they put the two together, and those sticky ends pick up that other enzyme. So that's how it's the other strand of DNA, sorry. The enzyme cuts it, and then it picks up the other strand of DNA from the other animal, and we're able to get jellyfish DNA inside the pig. Or a mouse, or a cat. They've done it with a bunch of different test animals in a lab. So those pieces are called sticky ends. So the first step in actually reading DNA, though, is to cut the DNA into smaller pieces, so they're a lot more manageable. The next step is actually separating the DNA. And they separate the DNA through a process called gel electrophoresis. What they do is they take the DNA fragments within a pipette, and they insert it into a line at the top of a gel structure. It's kind of like Jello. It's a very thin layer, okay, kind of like a substance of Jello. and it sits within this block on a flat surface that we're then going to run electricity through. So we have a power source up here, and we're going to hook the negative end to the top. We're going to hook the positive end to the bottom. What that's going to do is send electrons going through the gel. And what that does is it allows the DNA to move along the gel at different rates. Now, which ones move faster and which ones move slower? Let's think about this. If you're in water and you're trying to move something through the water, if you have two objects, one is very large and one is very small, and you're trying to push them through the water, the easier one to go through the water is going to be the smaller object, and the harder one to push through the object through the water is going to be the larger object because of the friction against the water. Same thing within the gel with our DNA pieces. The longer strands of DNA are going to have more friction, so they're going to move slower through the gel. And the smaller strands are going to have less friction, so they're going to move faster throughout the gel. Okay? So our DNA fragments start at the top, and you would put one person's DNA in each of these, so then you can compare them together. Send the electrons through, allow the DNA pieces to move through the DNA, and then we take and dye the gel piece. We stick it in a dye, and the only thing that picks up the dye is actually those DNA pieces. So with that, you get this strand and this picture of what's called a DNA fingerprint. And this shows us and allows us to compare people's DNAs. Longer pieces are at the top because they're harder to move through. Shorter pieces down near the bottom because they're easier to move through. And what happens is you can then compare the strands, because if you use the exact same restriction enzyme and you have two identical copies of DNA, what's going to happen is you should have the exact same DNA fingerprint because it should get cut into the exact same size pieces. So you can then compare multiple sets of DNA to figure out which ones match. And they do this a lot in crime labs. If they have a suspect and they have a DNA from the crime scene, they can compare them and figure out who it was who committed the crime by matching the two DNA fingerprints.